All right, let's jump right in. Welcome to The Explainer. I'm super excited to have you here today because, honestly, we're looking at a story that represents just a massive leap forward in AI news. For the longest time, we've kind of just looked at large language models as, helpful assistants. They draft an email, maybe help debug a script. But today we're looking at a groundbreaking case where an AI model, specifically GPT-5 Pro, shifted from being a simple auxiliary tool to an active, brilliant scientific collaborator. We're going to look at exactly how this AI stepped into a high-stakes medical laboratory and literally cracked a code that had stumped human experts for years.
Okay, let's dive into this. How do you actually solve a 3-year-old scientific mystery using completely unpublished data? Our story centers on an immunologist named Daria Unutmaz. For 3 long years, Unutmaz and his lab team were staring down this incredibly frustrating, totally stalled experiment. They had a puzzle regarding the behavior of T cells that they simply could not solve. They had the raw data, they had top-tier expertise, but the actual biological mechanism at play was hiding right in their blind spot. Traditional methods had just hit a wall, leaving them with a complete mystery locked inside a mountain of totally unpublished data.
So, to really understand the roadblock here, we need to look at what was actually going wrong in the lab. As we know, T cells are absolutely central to the human body's ability to fight off illnesses, right? Especially cancer. But in Unutmaz's experiment, when these defensive T cells were exposed to a very specific compound, they didn't do what was expected at all. Instead, they overwhelmingly produce cells that are involved in the body's inflammatory response. It's like they just went completely off script, and this unexpected detour halted the research in its tracks. Why on earth were the T cells reacting this way? The lab spent 3 whole years trying to figure out that logical link, totally to no avail.
And the specific compound causing all this chaos was deoxyglucose. It's a compound that directly influences the metabolic pathways inside immune cells. Unutmaz knew that introducing deoxyglucose was throwing a massive wrench into the T cell's normal operations, triggering this huge inflammatory response. But the exact mechanical pathway, the actual how and why, that remained a complete, utter mystery.
Enter GPT-5 Pro, analyzing the unpublished dataset. Faced with a complete dead end, Unutmaz decided to try something a bit radical.
Instead of just waiting for some sudden human epiphany, he turned to artificial intelligence. But, and this is key, he didn't just ask the AI a generic question. He took his laboratory's structured, raw, unpublished experimental results and uploaded them directly into the GPT-5 Pro model. So the crucial point is this highly straightforward, high-speed data-to-inference pipeline the AI followed. First, it ingested the structured experimental results. Second, the model mapped out all the complex variable relationships within that super high-dimensional data. Third, it flagged a highly specific correlation between the deoxyglucose exposure and altered protein dynamics. And finally, step 4, it pointed directly to the causal driver of the inflammatory response. Boom. In a matter of moments, it processed what took years to manually review.
And that causal driver? It was a protein called IL-2. GPT-5 Pro identified a previously overlooked mechanism. The deoxyglucose was directly interfering with the construction of this IL-2 protein. What's so fascinating here is that this specific IL-2 interference mechanism was sitting just outside Unutmaz's immediate area of expertise. And, as any researcher will tell you, the borders of our specialized knowledge are exactly where our blind spots form. But the AI didn't have that blind spot. It provided a retrospective, logical link that the lab simply hadn't identified independently.
From analysis to predictive simulation, stress testing hypotheses in a single interface.
Because let's be real, generating a hypothesis is one thing, but actually proving it is a whole different ballgame. Unutmaz wanted to test the model's reasoning to see if this IL-2 hypothesis actually held water. So he posed a massive challenge to GPT-5 Pro. He asked the model a counterfactual question. He basically prompted the AI to reconstruct the experimental conditions in a simulation and project the downstream effects of this exact biological interference. If deoxyglucose really was interfering with IL-2, what would happen next? Could the model accurately project these complex biological effects without ever running a physical wet lab experiment?
The answer was a resounding yes. GPT-5 Pro correctly predicted a massive boost in CD8 T cells' cytotoxicity. Put simply, the simulation output matched the expected biological outcome perfectly, an enhanced ability for these specific immune cells to kill lymphoma cancer cells. The AI didn't just find the broken link, it simulated the entire chain reaction and predicted exactly how these cancer-fighting cells would behave down the line.
Now, what's really interesting here is this quote: "The model couldn't have gleaned the results from the internet because they weren't published."
Honestly, this is the most mind-blowing part of this explainer. The AI did not just use a search engine to find the answer in some obscure medical journal. unpublished literally had not published any of these results yet. This means the model derived the outcome purely from the uploaded experimental context. It used true deductive reasoning on a completely novel dataset rather than just regurgitating existing literature. No way it could have just memorized this.
Which brings us to the technological paradigm shift, compressing the hypothesis validation loop.
Let's zoom out from this specific immunology case study for a second, because the macro impact on computational biology infrastructure is just staggering. We are looking at a fundamental change in how scientific discovery is paced and executed, and this brilliantly illustrates that shift. Look at the old method. Researchers would have to wait for peer-reviewed validation, build custom NLP pipelines, or run endless, tedious wet lab cycles just to confirm a hunch. Now look at the new method. You upload raw structured results, immediately request mechanistic explanations, and run counterfactual simulations right there in the interface. It completely compresses the hypothesis validation loop, saving incredible amounts of wasted reagents, time, and personnel hours. And the implications extend far beyond just immunology. Any scientific field that relies on complex, high-dimensional biological data is about to get a massive speed boost. Genomics, proteomics, metabolomics, all of these fields can benefit from this kind of AI-driven pattern recognition. Think about it. When an AI model can surface a plausible biological mechanism in a few hours instead of months or years, the entire pace of human discovery accelerates like wildfire.
Let's move to our actionable takeaways and see how this builds into best practices for AI-augmented discovery.
Because as magical as this all sounds, the AI is not replacing the human laboratory anytime soon. Researchers and developers need to drastically adapt their workflows to properly harness this new capability without falling into traps. The golden rule is this: treat large language models as high-speed hypothesis generators, not final validators. The GPT-5 Pro case was a success because the uploaded data was clean, well-structured, and incredibly context-rich. That old computer science adage, garbage in, garbage out, yeah, that still applies 100% in biology. The model's predictive accuracy is completely dependent on the quality of the context window you provide it.
So how exactly do you prepare that context window? If you are a developer or researcher building these ingestion pipelines, you must follow these exact data requirements. You have to normalize your data sets before prompting. You must ensure metadata is complete. Biological variables need to be clearly and accurately labeled, and your infrastructure must support structured uploads and strict version control. Basically, clean data is the absolute prerequisite to getting accurate, reproducible mechanisms from an AI. Furthermore, reproducibility is literally the bedrock of science. When AI assists in discovery, you have to document your work meticulously. You must record the exact dataset version you used, the exact upload format, the exact prompt structure you used to query the AI, and all of your simulation parameters. Why? Because other labs need to replicate your analysis. They need to verify if an AI's proposed mechanism, like our IL-2 interference, actually holds true across different cell lines or totally different experimental conditions.
And for the developers automating these workflows, you absolutely must implement critical validation checkpoints. Here is a solid safety sequence. Step 1. Automatically cross-check any model-suggested mechanism against known biological databases. Step 2. Ensure the system flags any low-confidence predictions. And Step 3. Always require a human expert sign-off before any AI hypothesis gets deployed into an expensive wet lab experiment. The AI suggests, sure, but the human verifies.
Which brings us to our final, provocative thought for this explainer. We've seen how GPT-5 Pro reasoned through unpublished data to solve a 3-year mystery in mere hours. If a model can surface these plausible mechanisms almost instantly, does the research bottleneck now shift entirely? Are we moving away from the slow grind of discovery and shifting the burden entirely onto human imagination in designing the right experiments and asking the AI the right questions? It's a completely fascinating paradigm shift, and one that will definitely redefine human-AI collaboration in the sciences. Thank you so much for joining me, and keep questioning.